just give it a couple minutes and let people in. And then we'll bring on Dr. Rommens to do her presentation. Okay, Nicole, I think what we're going to do is we are going to go ahead and we're going to start over. So if you are just now new here, let's see. Nicole, do you want to go ahead and take it from the top?
Thank you for your patience.
Yes, welcome to SDSF Live. Please let us know where you're logging on from. SDSF Live, take two.
Take two.
SDSF Live dose. So SDSF Live is brought to you from the Shwachman-Diamond Syndrome Foundation, providing support to families around the world. This show is public. Please do not ask or share personal information that you are not comfortable sharing. These shows serve as a library of topics on YouTube for families to access later, so it's forever imprinted. I am Michelle. I am from Michigan, and that is Nicole. Nicole's from Texas, and Nicole, I would love it if you could tell the viewers at home that who might not be familiar with who this legend is, who we have on tonight.
Can you hear me?
Yeah.
Okay, sorry. Dr. Johanna
No, we're doing
Rommens
great. Take two.
Okay, is a world-renowned genetic researcher based out of Toronto, Canada. She is credited for identifying the Sbds gene in 2002. Dr. Rommens also created the first mouse model with SDS genetic mutations. It's our honor to have her on tonight. Do we have Dr. Rommens?
We do.
Hi, Dr.
Hello,
Rommens.
welcome.
Hello, everybody.
Yes, take two. Try again.
Take two. Okay.
Number two.
We're going to turn the floor over to you.
Absolutely. If you want to go ahead and try to share your screen, Dr. Rommens, and I can allow it on up.
Let's try. I had it work for a fleeting moment a second ago, but I'm not sure.
We will give it our very best shot.
Right. Yeah. It's not.
I think we just had to give it just a second that time before when it was working, and then we snapped our fingers. And Dr. Rommens, while we're waiting, do you want to give everyone a brief history of who you are and what you do?
Sure. So my name is Johanna Rommens, and I am not a physician. Actually, I'm a scientist, and I work out of Sick Kids in Toronto, Ontario. And I've had long interest in genetic disease and in studying, what the causes are, what the genes that are responsible for diseases are. And I've had a long interest in Shwachman-Diamond syndrome that came out from very early times. I had worked on the cystic fibrosis gene. And then because there were some aspects of cystic fibrosis that overlap with Shwachman syndrome, I learned about Shwachman syndrome, but I also learned very quickly that very few people knew very much about Shwachman-Diamond syndrome. This was actually really long time ago now, before the foundation was as solid a group as it is now, and when it was just starting, when Joan Mowry was wanting to set up the foundation and already thinking about doing so, and then I got started. Then I got started into working on Shwachman-Diamond syndrome. Okay, now I still don't think I can share.
I think we had it. I'm gonna wait to see if it doesn't come up down here in the preview, where if we give it a second here and then we put it into the stream. I don't know what magic happened last time, but I would love if that magic could happen again. However, it needs to. just going to go ahead and keep it down there in the corner and see if it doesn't come up in a second.
Yes.
Did we click entire screen or full window?
I thought, well, I've been trying both.
Okay.
I recall we did window, and that's what works.
Okay.
All the times we tried it in practicing.
I know, we practiced it so much, and then it worked once, and then everything fell apart again. So, okay, well, why don't I do this? Why don't I try to share it?
Okay, that's what I
on
think
my
we need to
end.
try that, because
Yes.
even though it seems to be okaying that I get control, it's not wanting me to do steps that I can do to get my slides on the screen. So.
All right. So, Nicole, that means I will not be able to see to field questions if you just want to field them as they come in. Okay. All right. So let's do this.
Yeah. Okay. Okay. So we've got the live screen now, right?
Yep, we have the live screen. I had myself muted. Sorry.
Okay.
I will leave myself unmuted. There we go.
Okay, so what I wanted to talk today a little bit, and I'll try to put this, I tried to put this into a story, although I did get a lot of questions from several people. So what I wanted to do was try to have a bit of a narrative and answer your questions. So I try to put it together. So I think one of the things that I wanted to, or an aspect that we want to talk about, is how one goes about studying a disease gene. And some of this, I put this into a narrative, so I thought it would follow and make sense for everyone. Just so you'll see where I'm getting to, and discuss a little bit of our own research, but then also where things are going, where things are at, and what we need to try to achieve as things move forward in the fields. So the first thing, of course, we'd want to know about a disease is what goes wrong, why, when do things happen, where and when, and where and which they do. So if you're thinking about Shwachman-Diamond syndrome, you think, well, which cells and which organs? And as we know, there are multiple organs affected in SDS. Perhaps most concerning are the hematology, so the blood aspects of the bone marrow that we're concerned about, but there are other organs as well that are also affected. So we want to learn about where do they begin and what's going wrong. We also would like to have a picture of where things are going. That is, so if disease starts, and I'm talking about being very young, and it presents in this way, does it really stay the same all the way through disease, and as that individual ages, grows up and ages and so on? So these are all questions that one wants to ask. So how would you answer those kinds of questions? Next slide. So I think what one would do is, first and foremost, of course, you'd always want to be able to study patients. And this stays, I think, the first avenue of research. They need to be ongoing. And we already know all the amazing things we've learned about Shwachman-Diamond syndrome by looking at patient and looking at their clinical records and looking at questions specifically asked about patients. When you see something is different, we ask the question, well, why is it different and what's going on? So of course, it's asking about clinical records, but you also want to be able to look at patient samples, such as blood cells, or other bone marrow cells that some of you have all donated for research. So these are questions that you're going to ask. We know of a lot of series of studies that have been done from around the world, many countries, early days from Britain, from many countries, and even in early days, but now more recently, larger studies of registry data and such from around different countries. And I'm just going to pick one very recent example, which I think is a very elegant study, and that was the study that was reported this year in Nature Communications, describing the genetic pathways that were observed in looking in the clonal hematopoiesis of Shwachman-Diamond patients. And I know that many of you contributed as members of the registry. That study involved a study of well over 100 patients, and that included mostly patients from the United States, but also from other places. But it's a very elegant study, and that was the study Kennedy et al. As I said, that was reported. So that's just one example. So next slide. So other kinds of studies, though, that one might want to think about are going to be more cell and lab-based studies. And the first thing that one could study would be some aspects of the causal gene or the causal genes, since we detect that they're defective in patients. So we'd say, we'll ask the question, okay, well, how do they not work? And probably some of the neatest things that have been done, I think at least, have been to look at the structural studies of SBDS, the major cause of Shwachman-Diamond syndrome. Not in all cases, but in many cases, we find deficiencies in that gene, SBDS. So what does that gene do? Well, we first had to do a bit of work to figure out what it does. And I would dare say we still haven't explored all of that. We know quite a bit now, but not everything about it. And shown here in the lower right here is a nice image. This is an image from Dr. Alan Warren's website looking at SBDS. And SBDS is the purple or the magenta portion of that picture that's shown. This is a full space filling model of, in fact, the background, that pale blue is this, what's called the 60S ribosome, and I'll come back to that in a moment. And you can see the partner there, the blue one, the EFL1 blue protein, and also EIF6, the yellow protein that's present. So we're looking here at a real picture, and they've done some space filling with computer to make it look pretty and to show how tight things fit together of the SBS protein, as I've said, in purple, sitting where it works on the ribosome inside the cell. So that's some work that one can do. One can do that in the laboratory. One could do that using extracts from cells, and we can do them in totally in almost synthetically, in the sense that we don't actually need patient samples to study what does SBDS do, because we found this gene in almost all living species. So we can have multiple sources for the source of that particular gene. We can also make the protein because we have some tools in the laboratory with which we can do that. So this is one important type of study that is ongoing and looking. And as I said, the picture that I've shown here is a very special picture using probably one of the latest, coolest methods in biochemistry right now, a technique called cryoelectron microscopy, being able to see a picture and be able to see what it looks like really up close. So other kinds of work, though, that one can do to do research, next slide, would be to also think about models. And these would be either cell models. Now, in a way, cell models are going to be limited in some ways to source type in terms of what's available. So if we wanted human cell models, what cells can we use from the human body? Well, we can use, in theory, any cells, but, most of us want to keep our cells, so not every cell type's available. It's true that blood cells can be extracted and used and grown. Some bone marrow cells can be used, extracted, and grown, and these can be used. And more recently, there's been some methodology involving very elaborate way to take mature cells and readapt them into being stem cells. And you may have heard a little bit about these called iPS cells, and there are some people using those models. In their models, so they are cell models. When you think about it, though, from one aspect, in terms of thinking about what cells and what cells do, so cells make up your organs, and your organs have very different tasks to do. What is informative to understand about a disease is how cells talk to each other, how they comprise organs, how the organs talk to each other. So for a lot of diseases, what has been immensely useful has been to study rodent models, usually mouse, not always mouse, but usually rodent models. And there's many advantages, because if you have an entire animal model, you have a situation where you can look at the interaction between the organs. You can look at what happens if this fails or this part of an organ fails. You can see what's happening elsewhere in the body. And it's for these kinds of reasons, when we study a disease, a whole mammal model, a whole mammalian model is actually useful. And so that can be important. Although cell models as well can be important, in particular some of the newer cell models now that have stem cell-like properties, where they can take a stem cell, for example, and grow it into cells that form a mass that has lung-like function, or form a mass that ultimately can produce blood cells, or form a mass that can start beating like your heart and things like that. So those are some cell models, but there's also the animal models. And as I said, the animal models have the advantage of you being able to look at the whole individual. So for some aspects of the disease, that's an important part. Now, a really important part about any particular model is, and this is the big assumption that one makes, hopefully it's based in rehearsed in knowledge, and that's that you want the model to reflect the disease state, or at least reflect the disease state adequately for the question you're asking. But of course, you can ask many types of questions, and to get your answer. So you do make the assumption that the model will reflect the disease state. And sometimes usually you test whether that's true first before you proceed like with a big, long multi-year experiment or something like that. So that's important to remember. So we'll come back to the mouse models. Next slide. So let's think about how we would make a mouse model for SDS. So what do we know? Well, we know that mutations in SBDS lead to SDS. They lead to most cases. So it would make sense to make a mouse model that is carrying an SBDS gene that looks like what's been seen in an SDS patient. Okay, and what do we know? Well, we know when we look at the variants that cause SDS disease is that they result in loss of function of SBDS. That's a feature of the mutations that are found that cause the disease. And I say that because there are other types of diseases where you actually gain function, but these lose function. That's what we know about SBDS. And that's consistent with SDS showing autosomal recessive inheritance. That's the genetic process involved in how you inherit this disease. We do know that there are no patients born that have two completely null alleles. And what I mean by a null allele, I mean where the gene has been totally knocked out. Okay, so that would be absolutely no SBDS. So there's no such case that we know of than that. So that means that in the disease state, we have a very low level, but never complete loss of SBDS. At least that's the scenario of what's been seen. So that's something to keep in mind if we're thinking about building a mouse model. The other feature that we know, and this is an important point, is, and this is a very valid concern, is how close will the mouse mimic the human disease? Because we don't know that coming in. If you just ask the question, you don't always know that. But one feature that we do know is that the mouse gene is very similar to the human gene. In fact, of 250 building blocks of the gene that builds SBDS, there's only 6 differences between mouse and human. So that's really quite similar. It's not really very different. And it's actually true that this gene is quite glaringly similar in almost any species you look at. And it's striking. There are other genes that we've studied that don't look near this close between human and mouse, but this gene particularly looks very close. So a good argument for why it would make sense then to prepare a mouse model. The other feature to think about is the technology used to build. How do we build an SDS mouse? How do we build a mouse that looks like an SDS patient? And I can tell you that was a task that we took on some years ago now. And in fact, we built 3 different types of mouse alleles. So, and that actually lets you build multiple kinds of models, because let me explain. So we know that we have some variants in SDS, but even though I said nobody is born with 2 null alleles, we do know that some patients have 1 null allele. So we made one version like that. Okay, so that was the first mouse model that we made. We also know that many patients carry missense mutations. So that was, and I'll show you an example in a moment. So that was an example to make where one building block has been replaced by another building block. The building block changes enough to affect the function of the protein that's encoded. So that's something to think about. And then finally, a model that we made, and this is commonly done now in making animal models for disease, is something we call the conditional allele, which I'm going to explain in a moment. And I'll bring this up. So we've made these 3 alleles. So we have 3 gene versions, if you will. That's what I mean by an allele. And I'm going to show you how we put them together. I also want to say that the technology for this efforts has changed dramatically over even in the last decade, it's changed dramatically. And we used older school gene targeting technology to make the alleles we made. But now you could use a newer protocol, something that some of you may have heard about, called CRISPR-Cas9 technology. And that's one of the uses of CRISPR-Cas9 technology, which I'll come back to later in a moment. But let's first go to how mouse models were built. The next slide. So, before I talk about the mouse models, I just want to highlight some of the variants that have been seen in the SBDS gene that lead to SDS. This is a list of some common ones. And shown with the arrows, the green arrows to the right, are actually 2 examples. There's the double arrow and the single arrow. I think one's blue and one's green. These are the common variants that lead to SDS. In fact, many patients have the combination of these 2 changes. I want to point out the top one, the one with the 2 arrows. The 2 arrows, that variant leads to a null allele. That is, it would lead to essentially no SBDS protein. Okay, so a lot of people have that variant, but as I said, they would never have 2 of that variant. Their second allele is commonly the next one, the next allele, which is commonly known as the splice variant. Okay, now, when we were first thinking about making mouse models, it made sense to think about, well, let's make the common ones. And of course, we made the null, the knockout. We made it a little differently than it's depicted here, but the net effect was the same. We make a knockout where we take the gene out. But the splice mutation at the time was very difficult to visualize how to make it. And that was because I mentioned it was really important to make the assumption that the gene copy would behave the same between mouse and human. And at the time, there wasn't the technology to make that very feasible. So we actually, as much as I regretted it, was not able to make the so-called splice variant allele in mice, because at that time that wasn't feasible. I'm going to come back to this because it now is to do it, or at least attempt it. Okay, it's still got some question, but it would be interesting to try. And I'll come back to why it might still be useful for some things. So next slide. So next slide is the slide that's going to explain the Missense variant. And I'm sorry, I don't have the pointer, but I want you to look at the R126T in column 3, the allele 2. This was a Missense variant. This is where the arginine amino acid is replaced by the T amino acid. And in fact, we saw it in a number of our families. It occurred in some cases together with the null allele, which told us then that the R126T is not a null because it occurred in patients. So we knew that that was an acceptable variant to make that was representative of disease. So at the time when we were thinking, rather than make the splice variant, we actually made this one. And we made this one for the reason that it was not amongst the most common, but we had several cases. We had very good clinical records of these cases, and we could see a very classical picture of Shwachman-Diamond syndrome, as you can see here with all the data to the right here on this particular graph. So it made sense at the time to aim for this one. So next slide. So then we thought about making a mouse model, and I'm not going to go into how to do that. It's very complicated. It would take a grad student quite a bit of time to actually make mouse models. But we ended up making 3, as I've alluded to. We made a knockout allele, we made a missense allele, and that would be that R126T variant that occurs in exon 3. That would be in the black box in the middle, targeted SBDS locus. Oh, I can't point. Just past the FLP triangle. That is where R126 would be in box 3, the black box that's labeled 3 there. That would be where that occurred. And then we also made something called a conditional allele, which I've alluded to already, which is commonly done. And that's depicted actually on the bottom horizontal black and blue line. If you look, you'll see those 2 little pink triangles that say LOXP. Okay, so if you combine that version of the gene with another protein, with a protein, and you can deliver that protein to whatever organ you choose, you actually can knock out the SBDS gene in that organ, and you can knock it out only where that protein is produced. So for example, I can take out the SBDS gene in the pancreas, as I'm going to show you only, okay? And I'm going to show you some data that we were able to derive with using that. And this idea of a conditional allele is commonly used in genetic disease because it lets you do a couple of things. It lets you control the timing of when you lose the gene, and it lets you control where you're losing the gene. So that's actually quite useful for certain kinds of research to answer specific questions about when and where do you need the gene. So that was the third allele that we made, and that was useful. Now, next slide. So when you think about, well, how do we work with these alleles? So these changes are made in the mouse gene copy in a cell line. It's a very special cell line. It's a cell line that when we inject it into mouse embryos, becomes a part of the mouse that we're creating. And in fact, just like in a family, we would breed a mother mouse to a father mouse. We would look at the offspring. An advantage of mice is you get maybe 8 or 10 babies in a litter, so you get a lot. But you get autosomal recessive inheritance, just like you get in humans. And you will get, of a litter of, say, 8 mice, you will get 2 mice affected with SDS or will carry the alleles that I mentioned. We can make combinations of the alleles that I mentioned. So, for example, we could make the combination of the null and of the missense allele, and that was truly an SDS model because that had been seen in families. That combination had been seen in families. We could make, and we could show, in fact, that when we use 2 knockout alleles, that is 2 null alleles, that one of the reasons we don't see them in humans is because that's not compatible with life, and that is a model that in 2 null alleles we do not see live mice born. And so that's consistent with recognizing that we never see that in humans either. We also had a challenge, though, when we put the R126 allele, the missense allele, when we made a mouse with 2 of those copies. And what happened there was those mice died shortly after birth. And so that was also a problem. So that mouse also died. However, when we put the R126 missense allele together with the conditional allele, we could have live mice, and we could then use that to do some of our studies. Now, we could study from our mice. We could study through gestation. We could do some studies, but then later on after birth, we could do the studies because of the conditional allele. So that enabled us to be able to use the mice for various kinds of things. So next slide. So what did we do? Well, one of the first things we did was to say, let's take a little bit closer look at SBDS. And shown here in the top row of black horizontal smudgy bands, they mean a lot to me, but I'm going to try to convince you that what we're looking at is SBDS, as is labeled there, and that's the actual protein. That's the protein that's being made either in the wild type case, that would be the ++ lane, that's being made in the RR, that's the mouse that has 2 missense alleles, it still makes protein, or in the mouse to the right of that, the R/- that has 1 null allele and 1 R126 missense mutation. So there's still some SBDS there. I don't have the proper quantitation to show you. It's actually a little bit less, as you might expect, because the null allele gives no protein. So that's actually a lower amount. And what this gel actually projects is what is shown a little bit on the right, that the protein resides in the cytoplasm, not in the nucleus. And that's based by comparison to the other proteins that are shown. Shown just below is actually EIF16, a protein that you'll recognize that is related to SBDS and works together. That gene actually resides both in the cytoplasm and in the nucleus, whereas SBDS is by and large in the cytoplasm. So that's one of the first things that we did with our mouse models. And that's just a cursory look. So in the next slide. So then we also wanted to think more about what does the gene do. And before I tell you a little bit about that, I need to tell you a little bit on the structural work and some of the work that Dr. Alan Warren had done to look at yeast gene, because we could study in the mouse, but it turns out yeast also have an SBDS gene that we can study and learn what it does. It was very clear that this gene has something to do with the machinery in your cells that build proteins. And so you have the gene that's defective. In fact, it's a gene that works, that when its protein is made, it works in building proteins from other genes, or for all the genes, including itself. So one of the things that you could do is ask the question, well, what do the ribosomes look like? And what I mean by the ribosomes, they're the machinery that builds the cells. An important part of the ribosome, shown in the lower part, as you can see, there's a yellow part and a green part labeled 40S and 60S. Those 2 components work together to give you a working ribosome, and that the working ribosome is called ADS. Not too novel. It's related to a physical property that the ribosome carries. And it's that ribosome, it's a huge working machine in the cell, and its job is to actually read messages and produce proteins. And where we know that SBDS is binding, SBDS is binding on the 60S ribosome, like I showed you in that space filling model, and it's allowing the 40S to approach the 60S, so you can make the ADS. And if you look in the top part there, I won't explain exactly how that works, but you can see 40S, 60S, and the ADS. And in normal cells, you should see a very nice, sharp, tall peak that says ADS. So, and that would be what we would predict. So this is the machinery that's making proteins in your cells. This is what we're looking at here. So next slide. So So one of the first things we did was actually start to look at tissues and ask the question, well, how do the ribosomes look in SDS cells from our mice? And what I'm showing you here in the lower part are the livers from mice that were made that are carrying 2 missense mutations. And I'm comparing them to the upper panel where you can see that very large ADS peak in the upper panels. And the top panel is from a non-carrier wild-type mouse. The middle panel is from an SDS carrier parent mouse or sibling mouse. And the bottom panel is from an SDS case mouse. So you can see, in fact, that the ADS peak is missing. And that's consistent with knowing that this protein is involved in helping make proteins, that there's a part, if we go back one slide, you can actually see that ADS peak is missing because now in the bottom part where to get the ADS, you have to add the 40S and the 60S together. We can see that's not happening, that's the impairment. That's consistent with the structure that's been shown. That's consistent with what we've seen in various models where there's failure in SDS. So that's what we could show. Now looking overall, we could go ahead now 2 slides. If we look overall, what do we see when we look in our mice? So we're going to look now in the liver and the bone marrow. Liver is on the left, bone marrow is on the right. And if you compare the bottom panels to the top panel, let's just look at the liver for the moment. The bone marrow shows something very similar. And you can see the yellow arrows. If we look in the yellow arrows in the top, you can actually see sort of that dark gray staining matter around the red. If we look at the bottom, these are granulocytes that we're looking at in the liver. And just to remind you that in a very young liver, the blood cells are coming not only from the bone marrow, they're also coming from the liver. So this is pre-birth. You can actually see, but you can see the granulocytes are way down in number because there's much less of that dark staining gray. It takes a little bit of a while to get practice used to seeing these kinds of pictures, but we can see that there's deficiency here in those reforming of the granulocytes. And that's true both in the liver and also in the bone marrow. So that's a problem and something we're learning about when and where things happen. This is at this stage is just prior to birth. So we know at this stage, SDS is already presenting itself, at least in these particular organs. Next slide. So if we look at the same time points and we start to look at, and you all know about blood cells being low, if we compare here the different cell types, these are precursor bone marrow or blood cells, various types. You can actually see that when we compare the 2 right bars to the left bar in each of those sets of 3 bars, you can actually see that the right 2, these are the SDS representatives, the missense, and the missense null, as shown on the right there. You can actually see that the cell numbers are always lower than in the wild type mouse. That is the mouse that doesn't have SBDS mutations. So you can see that this is a difference. This is kind of an expected difference because we know this happens in people, but one of the first, or some of the first things you have to do when you build a mouse is you have to show that you're getting the same picture in the mouse as you're getting in patients. So this was an important experiment to show that. Now in the mouse, we can do a lot more experiments to look at this a little bit more closely to say, at this time point, we can see a difference. We can ask the question, when does the difference start to occur? Does the difference persist throughout all of growth and throughout all of adulthood, for example, and things like that? So that's just one little picture of one little experiment, but one could do many, many more to learn exactly when and why and where does the disease manifest. Next slide. So this is looking before birth. So the other organ that you recognize, or another major organ that you'll recognize that is affected in SDS is, of course, the pancreas. But an interesting facet when we looked in our mouse model at birth, and even though we saw all the problems in the blood and in the liver, the interesting thing is we don't really see too much wrong in the pancreas. And I'll ask you to take my word for it, but this left pancreas here, which is that of a wild type, looks very similar to the one on the right. And the right is an SDS pancreas at birth, okay? So we don't see anything. And I mentioned that you need to learn about when and where do you see disease. And it's not going to be the same time for all tissues, as we've learned, because here the pancreas at birth looks kind of okay. You see nice cherry red cells with those little blue dots. It's exactly what you'd want to see. You see that that middle part that you're seeing in the right with the dark blue specks are actually an islet. So those are the cells making insulin. They look quite fine when you compare the two. And we're looking at different scenes here, so there's very subtle differences, but overall there's not much difference. But we wanted to ask the question because we do know that in CF and in SDS patients that there are deficiencies in the pancreas. So we looked a little bit later, and this is now when we used our conditional mouse. So we had actually the mouse allele, which took out one allele, but the other allele would be the R126 allele. So the next slide. So we're looking now a little bit later. In fact, we're looking only at 6 days at P6. So this mouse is only 6 days old. And the mouse pancreas grows a little bit different than human. The human starts off, once birth starts, it starts off slow growing. It turns out in the mouse it actually grows very quickly. Within a week, it grows quite quickly, in fact. But one of the really obvious things that we could see, even though at birth it looked absolutely normal, you can certainly see when you look at these two pictures that the left and the right look quite different. And the most obvious things is that the right-hand panel is missing all of those dark gray spots. And that's very clear. Now, the dark gray spots are actually what we call zymogen granules, and they're actually the granules that contain digestive enzymes. And you can see here in this SDS mouse, even though the pancreas cells look okay, they clearly are having trouble making the zymogen granules. At least they were way down. And I'm just showing you one representative picture here, but we did several in many mice, and we looked at different time points and so on. So we could see that quickly one sees after birth, quickly one sees the loss of the digestive enzymes, even though the tissue looks okay when we look at it with different techniques. So the tissue is a bit sick, but not so sick it looks distorted. It just can't make proteins. Again, consistent with the idea that the role of SBDS is in making proteins. And the pancreas is, in fact, a very special organ that has to produce very large amounts of protein. Your digestive enzymes, they really produce a lot in a very short period of time. I've read that of all your organs, the pancreas in terms of per minute can actually produce typically more protein than almost all your other organs except for mammary glands, because mammary glands produce milk, and they're also very efficient at producing protein, but so are the pancreas cells. So this is something we learned from this. Next slide. So we looked a little bit later as well, and we actually started to see some concerns later as well. Now, it's always very interesting. This is looking at 20 and 30 days old. It's a little bit tricky to always parallel human growth and mouse growth because their lifespans are so different, and also the maturity of the organs is a little bit different. But there are people that are very expert in these in terms of being developmental biologists, where they understand how organs grow. And certainly we're looking here at day 20. This is post sexual maturity. This would be a teenage year, or only 20 days, but it would be comparable to a human teenage time. P30 is well into adulthood. And you can actually see the lower two panels. You see a lot of distortion compared to the upper panels in each of these displays. Particularly if you look at the right in the pink, you can actually see that nice bright pink on the top is not happening in the SDS samples, or at least it's no longer happening. And those very nice, robust cherry red cells are gone. At least a lot of them are now missing. They don't stain near as well. There's a lot of white, what appears to be white. In fact, we've tested this. It's not shown here, but that's actually fatty replacement. And that's consistent with what we know about patients having a pancreas that stays small and becomes quite fatty. We also see something called a process called senescence happening with age. And that's what's depicted here in the blue staining. We see almost no blue staining in the top two left panels, but we do see a lot in the lower two sets of panels. So this is a concerning thing to see, because it's a suggestion that this tissue is very stressed and very strained. And so this is something that we've seen and continue to actually look at to try to discover a little more. So something interesting that we're seeing with our mouse model, it's actually very difficult to do with human patients, is we've seen that the deficiencies of SDS actually present at different times in different organs. And I've alluded to that before, because that's something you can help figure out with a mouse model. And that's not something you can do by studying cells alone. So I want to highlight that's something you can do with a mouse model. So next slide. So the other thing you can do is you might see also things you're a little bit surprised to see. And this is, in fact, looking at the developing brain in the mouse. So the upper panel is the non-SDS. The lower two panels are SDS. We can actually see striking differences. And I'll draw your attention in particular to the last two panels, the one called tunnel, the bright blue one, the very bright blue with green glowing, and also the far right one that's labeled with the brown, and you can see little brown specks. And you can see actually two things here. These depict two things. In SDS, we see more bright green glowing cells in the center panel, and we see less brown cells in the center panel if you compare it to the upper panel. And you can see that very clearly. And this is telling us that two things are going on, that while the brain is growing, some cells are dying, and that's what the green is depicting. And some cells are not growing very fast, and that's what the absence of the brown staining is showing. Okay, so there's two problems in the developing brain, and we were a little bit surprised to see this so glaring at this stage. This is a very early stage. This is even now at a stage early in mid-stage gestation that the brain was having trouble growing. This is a particular time where the brain should be growing quite quickly, and it's actually having some trouble here. And we were a bit surprised to see how striking this was in the mouse. So this reveals another feature of being able to study mouse models. Not only can we learn when and where things go wrong, what is going wrong, we're also seeing a different process of tissue failure, if you will. That bright green is an indication of cells dying through a process we know called apoptosis. That's very different than senescence that we saw in the pancreas. So it's telling us not only do we got to worry about things happening at different times and different stages of disease, there's also different things happening in different cell types. So this was something else that we've learned. So what I've shown you here is some examples of what we can learn using mouse models that are very difficult to study in human tissues, just because in the mouse we would have access to any tissue that we need. So that's an advantage. Next slide. So as I've highlighted now, so what goes wrong, why, when, and where. But now if we want to talk about treatments and things like that, and how to have benefit, well, some of the things you need to think about, and this is how an animal model can be quite useful, is, well, if you have a therapy and you expect some benefit, how are you going to administer it? Where are you going to administer it? What are you going to measure? What timeline are you going to measure? And when do you measure? And what I want to highlight here is that by having a mouse model where you could look at, where we know when and where things happen, you have already the framework for thinking about, well, how do I have to administer the therapy? You can envisage that we would like to administer the therapy before super bad things happen, or super unexpected or unwanted things happen. That would be the optimal. But we also have to think about where and when. And it would generally be true in genetic disease, because we are talking about deficiency in many cells, that we usually don't talk about cure. And I know there was a question about that, and I know that different people have different takes on that. In genetic disease, we don't usually talk about cure, but that doesn't mean we can't tackle some of the problems in some of the organs, the most serious problems, such as, for example, if we can treat the blood, for example, in SDS, which would be very desirable because of the consequences of having, low neutrophils and things like this. We'd want to be able to treat individuals. Then we want to think, well, then we're obviously going to have to deliver a therapy somehow to the blood. And the blood's actually easier to access than most other organs. So that becomes feasible to think about. But these are the things you have to think about when you think about therapies and how to get them to work. And I want to highlight just how important animal models can be at giving you the platforms for how to think about how to administer, or even how to think about strategies for treatment. Next slide. So as I've already alluded to, there are many, many challenges in thinking about a cure or thinking about therapeutics, not the least of which is having enough information foundation on which you can plan something. You need to know what goes wrong to be able to think about how to fix it. But we also need a strategy, as I said, for which cells and which organs. And to remind you that developing therapeutics is a very intensive goal. It's very intensive because frequently there are many, many steps that have to be tried and retried. and retry it again. And often one starts out doing testing in cells, and that would be a common first thing to do. But then typically one goes through some processes where one also has to test animal models. You especially want to be able to test concerns of toxicity, concerns of off-target effects, and various other things that are concerning. And that would be an advantage of having an animal models. I also want to highlight for therapies, though, that there are many, many approaches, and you've probably heard a little bit of some of them. So one obvious approach would be to think about trying to develop a drug. But that's just one of them, and I'll come back to that point. There are also protein-based therapies, and if you think about it, some of you may already be familiar with some of them, such as Neupogen. Neupogen is a protein-based therapy. You're actually getting a growth factor for blood cells when you get Neupogen, and that's a protein-based therapy. Insulin that's given for diabetics is a protein-based therapy. But those protein-based therapies work because we know what the target cell is, and we know how to get it so that the target cells have access. So in fact, they're given into, the insulin is given into muscle, for example, and it gets absorbed into the blood where we need to be able to control our insulin. Neupogen gets into the bloodstream and is able to signal because it's a signaling molecule. It attaches to the surface of cells, and that's how blood cells, and that's how it works. So that has sort of a receptor or a receiver that we would recognize and we would know. But to fix SBDS, we'd have to get into the cell. So it's a little bit different question, a little bit more challenging, in fact, because we don't know how exactly to get it into cells. Now, there have been these ideas of gene-based therapy where we could direct a target or a drug, say, toward what we've seen the gene failing to do, or can we help? For example, if there's a little bit of gene product there, can we make it work a little bit better? So that would be one plausibility of how to develop a drug. The other plausible way to think about a drug and to think about SBDS, and this is the idea, I think, of Dr. Alan Warren. And remember if I showed you the structure about how there's a relation between the gene called EIF6 and SBDS. And we actually know that SBDS and EIF6 have to bind together. And if you don't have enough SBDS, you can't kick off the EIF6 off the 6DS. What if you could have a drug that enables that to work a little bit better, to say kick EIF6 off the ribosome without having SBDS there or not having very much SBDS there? So that's quite an imaginative approach. It's not going to be very easy to get that to work, but one could try and one could think about that. So I think my point I want to make is you have to have a fair amount of foundation knowledge on which you would build a strategy to think about treating and to think about developing a cure. But these are all approaches that can be tried. They're not going to be easy. They will take many stages and many phases of iterative steps where you're going to have to test and retest and be sure it's not toxic, because how much to give and how much to use and how much gets delivered. And, your liver is pretty suspicious. You take any drugs, your liver first tries to get rid of them really quickly. So you have to think about and worry about that and to deal with it. These are one aspect. Another aspect that, of course, has been discussed in terms of genetic disease have been the idea of genetic therapy. And that is, can we fix the problem from the beginning? The problem is in the DNA. We know that there is a mutation. Can we reverse the mutation in some way? And that has been proposed in some diseases, but it's proven to be very, very tough to be able to do that. Next slide. But I did want to bring up one method that has been proposed, and that has been the use of CRISPR-Cas9, which I've already alluded to. Because if we can make an SDS mouse where we make a change in the SBDS gene to introduce an SDS mutation, why can't we take an SDS cell and introduce the wild type and fix it? Because that would make sense that one should be able to try and to do that. And I can tell you this is a goal of a lot of people, and many people in genetic diseases are working on it because we're actually getting quite good at fixing cells. That is, taking a gene that is got a variant in it that causes disease. We can revert it back to wild type, and we're getting pretty good at being able to do that. But what we haven't been able to get very good at yet is, okay, how do we introduce it into an individual, or to which organs, or where? And how do we do it in that sense? One way we might be able to do that, and there's been a thought on this, and it's been thought of, is to actually, could we fix the bone marrow of SDS patients? And this is just an example, okay, of a suggestion of how to fix it. Take the blood cells out, fix the cells in the lab, put them back. And it would be kind of like a combination therapy of like a bone marrow genome therapy type idea. And I know that sounds really simple, but it's turning out to be really hard to do as we're learning. So that's not something that's immediate, but it's still an idea, and an idea that I'd like to see get encouraged. One challenge of that approach is that we're good at making correction, but one of the things that we're noticing is that sometimes we get off target effects. That is, not only are we targeting the gene we want to target, sometimes we hit other genes too, and that's not desirable. But I had read one very interesting proposal by somebody who says, well, and if you think about it, why don't we add, because one of the things you've got to do to fix a gene is you have to introduce some other proteins. And in fact, the protein that's why it's called CRISPR-Cas9. Knows that CRISPR and Cas9 involves adding a protein. The Cas9 is a protein. What you'd really want to do is have it enter the cell, do its fixing, and then get rid of that gene. So you want to kind of build a temporary gene. And one idea that I have seen is that somebody proposed to add the Cas9 as an RNA. And if you think about it, that's how the new vaccines have been made for the pandemic, and they're turning out to be really super good, quite safe from what we can tell. So can we introduce Cas9 as an RNA molecule into the cell? The advantage of an RNA molecule is that it has a very short half-life. Can we get it into a cell for a short enough time that it fixes the DNA, but then it dies because it degrades? And the virus vaccines are based on that principle, that you have the virus that simulates being infected by you have the RNA that corresponds to the virus. It simulates a viral infection. But then because it degrades quickly, you have its effect just long enough to get your immune system excited and for you to make antibodies so that you become immune to the virus. But then it fades away because the natural processes that degrade that RNA. And I've seen that somebody was proposing that for Cas9. And I thought, well, that'd be pretty interesting, because that might be a way that you don't have to worry about off-target effects of the gene being too active and starting to interfere with other genes besides the SBDS gene that you're trying to fix. So I think in the long run, when we think about therapies, there's not going to be one single therapy. They're going to have to be combinatorial kind of therapies. There's going to have to be maybe different combinations for different organs. That's something we're going to have to think about. And it will take a while to think about in terms of how to go about and do that. So I don't really have very much time, and it wasn't my plan to explain all of CRISPR-Cas9 technology. Could do a whole lecture on that alone. But if somebody was really interested, you could look here at Wikipedia Commons for a description of the latest new method of doing CRISPR-Cas9. Is something called search and replace genome editing, or prime editing. I've seen it called that, and that's been proposed as a mechanism to fix genetic or genes that cause genetic disease. So that's one mechanism, and we'll see. That doesn't talk about making the RNA Cas9, but that was a different article that I read. But it was an interesting idea and an important thing to think about. So I've tried to answer some of the questions, all but one. So let me go to the next slide, which I think is my acknowledgement slide. So I just wanted to, I will still, if I am allowed a few more minutes, I'd like to comment on one more thing, but I just wanted to acknowledge the lab members that generated some of those pictures that you saw of our mice tissues. I want also to acknowledge some work of investigators, which I didn't specifically cite when I was talking, because I think the neat thing about science is our data we build from each other, and we take some things for granted and we add the data all together. That forms our foundation for how we start thinking about therapeutics for a disease. And I also want to acknowledge SDS patients and families and their caregivers, and also funding agencies that includes the NIH, Canadian Institutes of Health Research, and also Shwachman-Diamond Syndrome Foundation. So there's one more question that I wanted to address, and that relates to building the mouse model for the splice allele, because I was specifically asked that question, so I'm going to answer it now. So I mentioned when we were first thinking about building the mouse model, and you remember here the two common mutations, the two with the double arrow and the one with the single arrow. So the single arrow is the splice mutation. So if we go to the next slide, so remember the 258 plus 2T to C, that is the lower line of text that you can see on this slide. So the very lower with that you can see, and on the right, the problem there is the alteration of the splice sites, which is to the right. And you can see, at least I think you can see, yeah, in the little red box, there's the GT. And if you go a little bit farther to the right, you can see another GT, except that now it's been replaced by red and it reads GC. Okay, so that GC is the splice mutation. And the quirky thing that happens when you carry the GC mutation is that your splicing of the SBDS gene will fail because your splicing machinery doesn't like a GC, it likes a GT. So, and that's a change that's happened, that's causing. Now, the funny thing is it doesn't like it, but it doesn't completely ignore it. So if we come to the next slide, and you actually look at what happens. So let's look on the top part. I'm just going to show you, yeah, the top part, you can see 1, 2, 3, 4, 5. So those are the 5 exons of SBDS. When those are read by the machine or by cell's machinery to code, the code gets read. It gets read into a protein, the SBDS protein on the right. You get this protein shown in blue. But if you look at the null allele, the C183, 184, that doesn't give you any of the blue protein at all. It just gives you a very short peptide that we presume degrades very quickly. That would be what we'd expect. But if you look at the splicing mutation, what actually happens is you get also that peptide, that short peptide that we presume degrades very quickly and has no consequence. And you also have a little tiny bit of the blue protein. And just to show you that, if you look on the picture on the right, I'm looking, and we look at the black bands about, 1/3 or almost halfway down in the picture. And we look at the lanes of the 2 black squares. This is a family shown above, the mother, the father, the circle is the mother, the square, the light square is the father. But the 2 individuals, patient 1 and patient 8 in this pedigree, in this family here, we're actually looking at their SBDS protein and some of their blood cells. And they actually carry a null allele and a splice allele. Okay, so they have the combination of the 2 mutations that are shown here. And if you really blow out this picture, which is what we've done, which is why things don't look that neat, but you can actually see a tiny little bit of SBDS protein there. So that's the little bit of protein that's there. And that's the consequence of the splice mutation. It leads to a little tiny bit of protein. That protein is wild type from what we know and understand. There's just isn't very much of it. There's not enough to prevent disease. But if you could envisage a therapy that would give you more SBDS protein, or that would make the SBDS protein work better, okay, so we're going to either have a drug, say, that somehow could make it work better, you could actually rescue this with such a drug, the situation, the splice by such a situation. So I know that there was discussion of making a splice model, and that was a model, as I said, we would have liked to have made. We didn't make it at the time because there was no easy way to do it in that. But there would now, with newer technology, it would be easier to do, to at least attempt it and to see whether or not you get the same effect as we see in human, where it makes a very small amount. Because then if a therapy was developed that could try to make more SBDS protein or try to activate what residual SBDS protein there is, this would be a good model to use to test that. That would be the scenario. So that would be an additional model to propose and to use for some types of planned therapies if they can be realized. So I just wanted to bring that out. So this would be how a splice model would be different than what is currently available and could have added value in terms of taking things forward. So that's how it's different from what has been made. The other possibility would be that the mice that we do have right now, as I've said, die soon after birth, is that they may live a bit longer because they do have a small amount of protein, but that's a possibility. It's not definite. The other concern would be is whether the mouse mimics the human in its splicing pattern. And there are a lot of similarities between splicing between human and mouse, but they're not exactly the same. So we'd have to try it to see if it worked. I think that would be the only way to figure it out. And so that would be, so this, I wanted to specifically address the question of somebody who would ask the question about what would the spliced mouse be. So that would be, as I've described, very similar to what we have done in the past, but now it's specifically going after this particular variant. But this variant does occur quite frequently in SDS patients. So that would be a desirable goal, perhaps. Okay, so I think what I'll do is end here, but I'm certainly happy to take any questions that somebody might have, either on what I've talked about, or if somebody had another question related to, say, genetics, I'm more than happy to respond.
I don't know if I've ever felt as much pressure with next slide, like hitting it too soon or too late. And then I was trying to follow you the best I could with my cursor. I'm like, I think this is where she wants. I think I'm going right here. And then you're like, no, I
thought you did fine. As far as I was concerned, but like I made the slides, so I remember exactly what's on them. But maybe if
you guys make
I learned a lot more than I think I would have because it was so interactive for me.
That was great. Okay, Dr. Rommens, we have a lot of questions. So just tell us, since we're going so far over, just tell us if you need to go. Okay, so we'll just ask and then you tell us, like, I need to go or take a break. First question, does getting a DNA test or genetic test help any patient figure out what medications are better for you? Would this help SDS patients?
So the person wanting the genetic test has SDS or not?
Has SDS.
SSDS. Well, currently, the advantage of having a genetic test would be that you've confirmed you have SDS. So in that sense, that's probably desirable and helpful. Usually the way you'd have to go about that is go through a genetics consultation, but usually a physician who suspects one has SDS would direct that. So you could get a genetic test in that sense. So in terms of having a genetic test, so if there's a positive genetic test, you have an answer, if you will, for what's wrong, and that's sometimes quite important. It's also important from the point of view of when a doctor is thinking about, okay, how does he or she help you manage your disease? So maybe there would be things for monitoring and things like that you would want to do to be very careful, as is typically done. If we look at the consensus document that talks about what should be done at different time points, when there should be a bone marrow check and when to check and how to check to make sure that nutrition is good and things like that. So those are all things that I think are a little bit less stressful if you know your genetic test results, in the sense that you have SDS, that a doctor wouldn't look further. Because if you don't have a positive test, I think a doctor would know that then, but he might also look further and keep asking questions. And the reason I bring it up is I know that when I've looked through the clinical records of many of our patients, that almost every Schwachman patient has also been checked for CF, for cystic fibrosis, because there are some things that overlap.
Right.
So of course, and CF is more common than Schwachman syndrome. So many of our patients I've seen have all been checked for that. And this is sort of a natural sequence of events that doctors want to do to know what's wrong, because then they know what to worry about at what stage. And so that's important to do. In that sense, a genetic test is probably very useful.
Is there any, I think what she's asking was, are there any tests for, Michelle, you can help me.
We've had a couple done for psychology with the spit to see what type of medications a child or patient responds to. And at least in our experience, they have come back completely bonky, wonker, all over the place. Medicines that we've tried, we have failed, came back that they should work, and we know they did not.
Well, maybe I'm not sure on what genetic test you're talking about. I mean, I know that there are some tests that can be done to see if you're allergic or reactive to some drugs. Like, for example, there are some tests that are done on some of the genes, and I'm supposing this because I'm not sure what genetic test here. I know that they would look to see how your liver reacts to some drug therapies. And there are responses to genes, depending on what makeup you have, that might decide what your dosage should be and things like that. There are some. I don't think of them in Schwachman syndrome, but I know for other diseases, they worry about that. They want to see how sensitive you are and how fast does your body turn over the drug. So they will condition the painkiller dose, for example, accordingly to how quick your body reacts with it. Or I mean, the problem is how fast does your body get rid of it? So, and that is quite variable between people. So that's something sometime they want to look. They also will look to see if there's concern. I know there's some tests they do to see how reactive you might be towards certain antibiotics and things. And there are tests for that kind of responses.
Thank you. The next question is, will you explain what the odds are of producing an SDS mouse that is exactly like an SDS patient?
What the odds are? Well, I mean, I can say 0 because a mouse is not a human. Like, you're not going to get an SDS mouse without whiskers, I don't think. Like, because mice have whiskers.
I would love to see it tried.
Well, I don't think so, because I think a mouse without whiskers is not a happy mouse. They like their whiskers very much. So it's very hard to answer that because, you know, the right model is the one that you need to answer the question you're asking. So that's why, anytime you make a model, you have that assumption that I've said, that the model is going to really model the disease, and we don't know that exactly. And we know of other disease examples too, where the mouse model has not well mimicked the disease. And I can tell you one of them is cystic fibrosis. The mouse model does not mimic the disease very well. Now, part of that is because, you know, some of the bigger problems in cystic fibrosis involve the lungs. But if you think about it, humans walk upright and our lungs, you know, are up. But a mouse is on 4 legs. And so the organ works differently. You know, so it won't tell you everything. Now, we've learned a lot about CF disease by using mouse models, but people will say, well, but so for some of the therapies, for example, they actually can use now some of the therapies they have now in the mice and show improvement, just like in people, but they have to be measuring slightly different things because we've learned that, you know, the gene does slightly different things in a mouse because he walks on all 4s,
right,
as opposed to standing upright. So not everything will ever be exactly the same. And you know, but you need a foundation of, do you recognize how it's different? And by recognizing how it's different, you still learn because you know how it's different, or at least you know, taken the efforts to learn how things are different.
Is it necessary to have a mouse model to do gene and/or drug therapy testing? Are there other options?
So right now, to do therapy testing, it is commonly requested by the FDA to have an animal model, and typically a mammalian model. It wouldn't always have to be a mammalian model. I think there have been some drugs approved without going through an animal model. Sometimes they want even more than a mouse. They'll want a model that mimics certain specific aspect of the disease better than a mouse model, and that's happened. Like sometime they usually now don't need chimpanzee models because usually we don't allow people to do that anymore. But they at one point wanted primate models, and there are still things where they will want a primate model only because primates look more like humans than mice, and so sometimes they would do that. There are some alternatives now. There are more alternatives than there used to be. So, for example, I didn't talk very much about, but the induced pluripotent stem cells, or stem cells that can be grown into sort of pseudo organs. Like you can, for example, you know, grow cells and they actually into sort of brain cells and connective networks, and they'll grow into networks, and you can actually show that the therapy could at least relieve some aspect of what's not working. It's not a whole animal model, but at least it shows you that it's effective at this specific step or this specific stage. So in that sense, there are alternatives. You have to know where you're coming from to know where you're going a little bit. but there are some, and that's something that there's a lot of work being done to try to improve that and to try to work on that. I mean, that actually had to have happened when they developed drugs for cystic fibrosis, like I said, because they couldn't use the lung mouse model. So they actually started growing human cells in a dish. And if I look back in the last 10 years, what we can do now in a dish is, a lot more than we could do 10 years ago. We didn't even know how to grow, and they're really hard to grow. I have to tell you, they're super hard to grow.
And stem
I can't
cells.
Now we can measure things. We can measure things that parallel lung function. So that's worked out. But we've also had to wait for that. And so it costs time, a lot of time.
Do you think that therapy for SDS will have to be addressed to individual organs?
I think probably, because the gene is inside the organ, like it's inside, it's not in your blood. It's not, it may have to be by organ. I also think that the blood is more accessible than the other organs. So one would think that the therapy for the blood could be quicker than the other organs, potentially. It's also the most concerning picture too. So probably that should get attention first.
Right. What is the average length of time it takes to develop a drug therapy?
Oh, you don't want to know. Anywhere from 10 years to 30. Long time. It's a long time. And I'll tell you why it's a long time. It's a long time because a little bit it depends on, okay, what are you trying to do? And how specific can you get it? How lucky are you? Because there's also a great deal of luck involved. It just triggering certain things. Like, for example, if we want to say, well, if we can overcome not having SBDS by changing how EIF6 works, okay, we have some rationale and some thinking for why that might be true. But if we make a drug that makes EIF6 work more poorly, because that would be what we want EIF6 to work more poorly, what happens to other cells that get it? Are we hurting them too? Are we making them work more poorly too? And that's why the drug could be tricky. Not impossible, but tricky. And we'd have to know how to measure that. We'd have to think about carefully how to measure that. Now, the good thing in biology is lots of things allow a lot of variation, which is a good thing, because maybe there's a range where you can tolerate it. Maybe you can rescue just enough to make the cells happier, but you don't impede other important things that they have to do in terms of other proteins that have to be made, or change. You don't change the repertoire of proteins that are being made, but it's something that we would worry about. But you got to try.
Got to try it, yeah.
You got to try.
Has this ever been successful in any other genetic disease?
Drugs, some of the drugs they have for cystic fibrosis are pretty potent right now. And I think that's probably one of the nicest stories. And they're doing different things. So some of the drugs now help a poorly working protein work better. Okay, it makes it work better. And that's why I mentioned that, because there is such a drug, and it works on some variants that cause CF. There's other drugs, and sometimes they're mixing them together now because you want to help them make them work better. But the other problem that we've seen the cystic fibrosis gene is quite different than SBDS. It's a very large gene. It has a lot of pain in getting made. The SBDS gene, there's generally a lot of it in your cells, but there's not very much CFTR protein, or the protein that's affected in CF. And one type of drug actually helps it pass through the cell in the right pathway. And then we add the other drug to make it work better. So some people are getting combination therapy, and they're both drugs. And those have been quite remarkable that we've seen develop over the last 20 years. They're quite remarkable now when you look at some of that data. So it's been very exciting in that particular field.
Yeah.
So Nicole, I'm going to jump down just to the end. We can backtrack, but this one was in response to one of Joanna's answers. Was Lisa said, you say 10 to 30 years, but has this development already started?
Yes, in the sense that the idea of, and some of the, for example, when we look at some of the modeling that Dr. Warren has done, to think about a drug and to think about how to make it, you have to have a very good picture of what your protein looks like, how a drug might dock onto that protein, how to look at it. So in that sense, like I said, that framework of knowledge that you need is already quite well done. Well, a lot of it has been done. And I know, and I think Dr. Warren is going ahead with some screens to start to look So what you would do is look through every chemical that we know. Well, not quite that, but that would be the kind of example that you would do. You do sometime, though, have to go through a multiple trial or multiple attempts. Like you make one drug and you think, well, the drug has this property, which is really good, but the liver doesn't like it and kicks it out really quick. So can I keep the same drug structure, but add one thing to make it bypass what the liver is doing? Because, the liver is a very suspicious organ. It just kicks everything out it doesn't like. If it thinks something's foreign, out it goes. But can I bypass the liver? And so these are why you need reiterative plans, that you start out, and what you start with, your end drug, it will often be quite different, but you want to retain the good parts of it, get rid of the bad parts, but sometimes you have to figure out what the bad parts are on the go, which is why it takes so long. That's a reason it takes this incredibly long time.
Can we throw up Dr. Corey's comment, just because it's such a treat to have Dr. Corey on here too.
I know. It's like it's 2 for 1. I love it.
I know. We weren't expecting
I don't know how much of it's going to show because it's very long, but I'll read it out loud. Yeah. Dr. Seth Corey says, we look forward to seeing Johanna's talk. We are working on a drug with our medicinal chemist at Cleveland Clinic. The average time for test tube to drug approval is more than a decade. But maybe we can do this more quickly with an orphan drug. We are working with Valentino
Bizzarri. Yeah.
Thank you. And Marco Spoli on another type of drug from their exciting discovery on how to make protein. Some might think it's just nonsense. No pun intended.
Okay, maybe I should explain that. Okay, so what Dr. Cipolla and Dr. Bizzarri has been working on, and I see if they're working with Dr. Corey, I get that because I know what each of them are doing. So what they've been trying to work on has been this idea, and this has been, I didn't talk about this because there's so many things I could have talked about. So one of the ideas that some people have had is, and I talked about this, that the cases of the variants that cause a premature stop, that is, they cause the protein machinery to stop. But there have been attempts, because these kind of mutations or variants cause problems in many genes that cause many diseases. So people have been trying to come up with a drug that perhaps prevents that stop from happening. So that's what Dr. Corey's pun on nonsense, because when that happens, we call it a nonsense allele. Mm-hmm. So can we override the nonsense allele is what such a drug would attempt to do. And they actually say they've screened and they have found a couple. They have some already. And these were screened for other diseases, because you have these nonsense alleles in many genetic diseases. And they have been tried. There have been several rounds of them. Some of the challenges have been is that they work in one case and not in the other, or they don't work the same in different cells of the same type of disease. So it's been really tough. It's been challenging, but these studies are ongoing and they're getting better at them all the time. They have done a trial in cystic fibrosis with one of these drugs. Did not work very well, but the trial was very heavily criticized for how it was done. And that was that this drug interferes with another drug that cystic fibrosis patients were taking, and they didn't account for that. So the trial was considered maybe not done in the best way. And I don't know if they're going to retry or not, or they're maybe waiting, because there are a lot of groups working on these type of drugs that may work, with the hope that they'll work for more than one genetic disease. And that's what Dr. Corey, and I thank Dr. Corey for bringing that up, because I didn't actually bring that up. But that would be another type of strategy of approach, but it would only work for individuals with the nonsense variant. But that would be more than half of cases with SBDS.
Next question is, do you need a splice model to test drug therapy?
So there would be types of therapies where the splice model would be useful. One of them would be a situation where, for example, we wanted to make the SBDS protein work better. That might be an example, because the variant that we have is got a mutation in it, right? It's got the missense mutation that I've described. We know that protein doesn't work perfectly. We could try to make it work better too, and that's how in cystic fibrosis the mutations in the drugs have worked. So it's plausible it'll work. But it might be better to have a little bit of wild type protein, and that would be what we would get out of the splice mutation. The other feature of the mouse model that would be useful was if somebody could target the splice problem and try to fix it with a therapy that fixes the splice problem in the splice mutation, would be then you'd want to splice mouse to try to fix. I don't think there's a compound that does that right now, but in that sense, the splice model would be useful. So like I said, the right model is what you're trying to fix and what you're trying to solve. So in the end.
What are the odds of making a splice model close to human?
So in terms of, from what we've seen from the models that we've made, I think the models mimic human reasonably well. In fact, if anything, I would say the mice look sicker than the patients. So, but they parallel quite nicely. The more severe, but they parallel. There actually are some other reasons why mice may look more severe than human, and that's actually because humans are what we call outbred and mice are inbred. So there is sometimes an advantage and a disadvantage of that in mice. But the other, I just lost my train of thought. The other thing I wanted to say, what was the other part of the question?
What are the odds of making a splice model close to human?
Oh, so closer to human. So in terms of what the disease will look like, I think the models already do look like disease. So I think the splice should look like the human disease. It might be a question to what degree. It would also be a question of how similar is the splice mutation recognized and responded to in the mouse versus the human. And that's something we don't know, although that can be tested even before you have the mouse. As soon as a version was made in the cells, and I don't know if this is in the plan or not, but I would recommend it, is that they actually start looking in those cells to see already, is there a little bit of splicing going on? Because all cells express SBDS. And I described to you, you make the allele first, then you take those cells and inject them into a mouse embryo. So you could already at that stage test, and you wouldn't even have to actually make the mouse yet. You would just take your mouse cells that have already had that allele made. So I would do it at that stage to test it, and it wouldn't be as like with the finality of what the mouse would be, but it would already be a stage that you could see. So I would take, very similar type, like a fibroblast cell or something like that from the cells that you have, and see, does it match what happens in human fibroblasts with that variant? And you could compare it. Okay, so that would be a very important step to do. I'm sure it's in the plan.
We only have 2 more questions for you. We're almost done. Next question. If the FDA typically requires a mammalian model, don't we need a mouse model with the splice mutation to move forward with the potential therapies for that mutation?
Yes, for that mutation, possibly. Although I have to say the FDA is quite not always consistent in exactly what they want. So it's good to be ready, would be my suggestion. They vary. And maybe they won't even want a mouse. They might want something harder than a mouse, more a bigger animal. I don't know. It would depend. It would depend.
How far out, this is your last question, how far out is Dr. Warren's drug from testing on humans?
Oh, I don't know what stage. Like he's at very early stage from what I know. I don't know exactly what he's been testing. I mean, I know he's looking for models and things like that to test, but you would go through many rounds of that, of improving the drug. It's not even really fair to call it a drug. I'd say it's a chemical at this stage, for which they're probably trying to, because usually when you develop a drug, you get a structure that looks like something is happening, but then you've got to go through all the toxicity concerns. You've got to go through all the side effects, the off-target effects that can happen. And what will happen is then the drug will have to get changed to alleviate some concern that you might have about toxicity. And this is what takes long. And there would be rounds of that. You would go through that too, many times, sometimes 10 times before in the end you actually have a drug you would ever, be ready to say, is it ready to try in humans? And the hard part is first thinking about, okay, what is my endpoints going to be? Because in a human, you can't tear their organs apart and things like that, right? Mm-hmm. You'd need to have something you knew you could measure and see benefit. And an obvious thing to look for, do the blood counts get better? I mean, that would be, but that's an easy endpoint, but many things go wrong before you get to that endpoint. So you would want to have tried many things in cells and probably mouse models as well.
Thank you, Dr. Rommens, for everything you've done for the SDS community. You are amazing. You're someone that I actually tear up when I see you at camp because you've done so much for my family specifically, and I'm just so thankful. So,
Well, thank you. You're very kind.
and you're such a kind human as well.
We'll keep at it.
That's all we have for tonight.
Okay.
We've had you for hours. Thank you so much.
Thank
So
you for
reminder,
having me with all the technical difficulties and.
It's good. It's head for the Olympics. It's 9 a.m. in Japan. It's almost soon to see what's happening there.
Oh, it's time. I love it. My kids have been obsessed with watching the Olympics, and then they went in the pool today, and then they were all practicing their handstands and their dives, and I'm not discouraging it.
That was fun.
Lots of thank yous coming in from the community.
Yes.
Thank you for all the work you've done. You are a true legend in our eyes. Thank you.
Well, you're most welcome, and thank you. We'll see you.
Have a great night. We have lots of shows on deck. Everyone have a great night. Thank you for hanging out with us.
Bye-bye.
Yes, Nicole and we have lots of shows coming up for you. We have, we're trying to get dentists. We are working on, I think, orthopedics.
Lots of hematology shows.
Lots of hematology shows. There's quite a few of those.
The
Maybe a cat show soon.
Back. The SDS registry is coming back. So we're working on lots of shows. So
yes, the registry is coming back.
We'll advertise those soon. Thank you guys for hanging with us with the technical difficulties. We appreciate it. And you just never know with these live shows. So
it's life. It's what happens. Like I've had a cat trying to jump on my keyboard for the last 30 minutes, and the only way I could make it not happen was shoving the cat on my lap. I'm like, can you stop? But no, really, we appreciate this.
Great night. Thank you so much.
Have a great night. Bye, everybody.